Okay, I think I can start with the welcome and introduction in the meantime. Again, hello everyone, and welcome to Emerge episode 16 with Maria Wahle. I'm Irishan Govender. I'm from the Resin Biosciences team, and I will be the host for this webinar today. Just some house rules before before I introduce Maria formally. The webinar is currently being recorded, and it will be available on our YouTube page in the coming days. So if you didn't watch it or if you want to rewatch it, you can do so there. After Maria presents, there will be a Q&A session. So I encourage you all to use the Q&A function or the chat box to put your questions in so that we can ask them to her at the end. Alternatively, if you would like to ask your question directly, you can raise your hand and then I will unmute your mic and then you can speak to Maria directly. So today we have Maria Wahle presenting some of her work. She's a PhD student in the lab of Matthias Mann at the Max-Planck-Institute of Biochemistry in Munich. There she works on immunopeptidomics, MS technology, and she does method development. Her talk today is entitled The Potential of HLA, Plasma HLA Peptides Beyond Neoepitopes. We're looking forward to your talk, and thank you for joining us for episode 16, Maria. So over to you.
Well, thank you very much for this introduction and also for the opportunity to present my workflow within this webinar. I hope you can see my slides fine, and I'll just dive right ahead into the topic of immunopeptidomics. And before I go too much into detail about what I'm doing, just a quick update to get us all on the same page what immunopeptides actually are. Immunopeptides are what is presented at the surface of nearly every cell in the body by the so-called HLA protein complex. And the peptides that are presented by the HLA protein are basically digested proteins, digested cytosolic proteins by the proteasome that are shuttled to the ER and then transferred onto the cell surface to be presented to T cells. What you see here on the left is the pathway for HLA class 1. So CD8 positive T cells are what these peptides are presented to, and mainly cytosolic proteins are presented. I'm not going to go into MHC class 2 or so-called HLA class 2, but focus on HLA class 1. What is really important about those peptides is that they are digested by the proteasome and selected by the HLA protein. So every one of us has optimally 6 different alleles of HLA proteins. So basically 6 different classes of peptides can be presented at the surface. So the presentation on the cell surface is guided by the HLA type and not by a protease, which is typically the case for proteomics experiments. As I said, there are 2 different classes, HLA class 1 and HLA class 2. HLA class 1 is usually endogenous proteins which are presented. The peptides are 8 to 11 amino acids long typically and have an HLA epitope. It's a bit different for HLA class 2. But again, what is important is that there is not really a pattern for digestion, rather a pattern for presentation. So the general challenges if we analyze immunopeptides is, especially human immunopeptides, is that there is limited sample availability. Especially if we want to look into healthy tissues, we can't really go ahead and take a lot of biopsies from healthy people to analyze what is presented there. So a typical source for HLA peptides are cancer biopsies. And so you don't have a lot to go on there. And the next point is these HLA peptides are usually analyzed before doing an affinity purification of the HLA peptide protein complex. And as most affinity purification processes need antibodies, this is usually connected to a high cost and really high investment for a single sample, which you can usually only measure once. The mass spectrometry specific challenges that you face analyzing those peptides is that you need to have an efficient way of measuring singly charged precursors. This is the case because, as I said in the beginning, you don't have a proteasome, you have the proteasome digesting the proteins and not a protease like trypsin, where you have a second charge from your from introduced kind of by how you choose to digest your sample. So you can end up having a lot of singly charged precursors, and mass spectrometers are usually not optimized or tailored towards single charged precursors. Another challenge is, again, the non-specific nature of immunopeptides, which really boils down to how you design your search in the end, which I will come into a bit more detail later about. So I'm working usually with immunopeptides that can be enriched from plasma, so with soluble HLA protein complex. As you might have saw in the first visualization in the beginning, on the right side, you had a small panel about sHLA proteins, so proteins which can be found in body fluids. There are different mechanisms of how they can be generated. There's a possibility of an alternative splicing variant, which doesn't have the membrane anchoring domain, or shedding from the surface, or any other way of generating those in body fluids. There's not a lot known about what is presented of them. What is known is that there are altered levels described in several diseases, such as autoimmune diseases or also cancer. And these basically serve as a collection of immunopeptides presented by a variety of tissues by just looking into the body fluids. So what I said earlier, that it's hard to look into actually what is presented on a tissue because we can't take biopsies, but we can easily obtain plasma. So what we designed, or what I designed in the first part of my PhD here, was the IMBAS-MS workflow. This is a workflow tailored to force efficient and high throughput enrichment and cleanup of HLA peptide protein complexes from body fluids in combination with a computational library-based DIA analysis pipeline, and later on interpretation of those peptides. And we called it IMBAS because it stands for immunopeptidomics by biotinylated antibodies and streptavidin. So if we start this workflow, I said it usually needs antibodies. I still use antibodies, but what is different for this workflow here is that the antibody is biotinylated and I capture it with streptavidin beads. And especially while analyzing plasma, this gives me the advantage of not needing a preclearance steps for usually IgGs in plasma, nor do I need to do a cross-linking of my antibody to the beads prior. So a bit more into detail here. What you can see is when you start with a typical sample, you can start with plasma, but you could also start with cells or with tissues. You have a crude mixture of the protein peptide complex with whatever proteins in the sample. As I said, I couple my antibodies basically to the beads by the interaction of biotin to streptavidin, a very specific and very strong bond. So I just need to mix this with my plasma. I don't need to cross-link the antibodies to the beads, and I also don't need to pre-clear the plasma prior, because what is traditionally to be done is either doing cross-linking step or using protein A or protein G to capture the antibody against the HLA protein complex, which gives obviously the problem in plasma that you have to pull down previously the IgGs to not pollute your sample. We can do this on an automated platform and wash and elute the peptides, separate those peptides from the protein complexes, and then I load the peptides onto Evo tips, which then can be later on measured on the mass spectrometer. To give you a bit of an idea how it looks like, so this is how basically the robot does the workflow. I take up the beads, I add them to my sample, and then I incubate them, and I basically can, here is a little demo about 48 samples, but you can also do it, of course, with a whole plate, transfer them. And what's really nice with using the beads actually from resin is that we can do it very fast and very clean without really having a lot of bead loss to the tips or bead loss on the plastic. And they separate really fast, they separate really nicely, and this allows us also to really do this on this robotic platform because we have a way of doing the enrichment without actually a lot of long incubation steps in between. So assessing this workflow, what was really important to me is to reduce the amount of antibody I need to do this, to really have a cost-efficient protocol in place, to have limited steps, to do it in one pot, and to have it easily doable by other people and other laboratory setups. I show you here a bit of an example of how I assessed the workflow in terms of how much antibody I needed. So basically in purple, you see an enrichment with 1 microgram of antibody. In blue, you see an enrichment with 10 micrograms of antibody and different plasma volumes in the bottom, different plate setups. And all those were assessed with a DDA on a 31-minute gradient and analyzed with the MS Tracker. And what is really nicely is that while increasing the volume, I still increase my depth, but also I have a high workflow reproducibility between different workflow replicates. So the workflow is very easy to use and gives us a clean result. So next step is really the acquisition. And I want to spend a few minutes on how I decided to use those methods and also on the mass spectrometer I used here, because those steps are pretty important, really important to take into consideration when designing and performing those experiments, especially if we later on want to apply it to a lot of samples or maybe only a few samples. So a lot of consideration goes into what kind of gradient do I use and what kind of method do you use. So choosing the gradient here for me was a compromise between speed and depth. So I compared all the gradients are basically run on the EvoSep system. So it's preformed gradients, it's also preset gradients. The part to decide on here really is the time, the gradient length, and the column setup I'm going to use. So these are either done on 8 centimeter columns, so the 21-minute gradient, or on 15 centimeter columns for the 44-minute gradient and 88-minute gradient. And EvoSep calls them usually with the term samples per day, which gives you an idea on how fast you can actually measure a high amount of samples here. And we compared the more microflow gradients, low microflow gradients, to the nanoflow gradients, the WISP gradients. And what we see here is that we get a nice increase in identifications from using the WISP gradients. And we chose the WISP 40 to continue here for time reasons. And these were done on the ion optics setup. Both again measured with a timsTOF and from 200 microliters of plasma. So going to the method design for the timsTOF, for people that might not be as familiar with the timsTOF system, the timsTOF is a TOF mass spectrometer, but up front of the TOF detector, it has those TIMS cartridges. This is another dimension of separation. In addition to the M/Z separation of precursors that are measured, you get the ion mobility separation. This leads to a bit different design of methods and also will end up giving you another layer of separation of your precursors. So basically just as a heads up. And if we now compare an immunopeptide sample towards a tryptic digested sample, we immediately see already in the M/Z dimension that the peptides we're measuring are distributed very different towards a tryptic digested sample. So on the top left, you see the tryptic digest, on the top right, the immunopeptides, and the precursor distribution looks completely different. This also translates into the ion mobility dimension. This is basically the y-axis here. And you can see that while the tryptic digest has more of a one homogeneous population here, the immunopeptide sample has somewhat two distinct ones. And this is really the single charge precursors that come into play here, what I said in the beginning about where you have to keep an eye on when you measure those. This also translates into how we can design a DDA method. There are other publications that go into more detail about this. I had it in the previous slide if you want to read up on those, but this also goes into how do you design a DIA analysis, a DIA method here. Because what we can do is we can strategically place those windows with a variable width, so DIA windows, and optimize these to immunopeptidomic samples, which are again vastly different from tryptic samples. So it is very important to invest those method optimization steps to really get the most out of your sample. And there's a nice tool available of those written by a colleague of mine. You can check it out in her publication or also on GitHub. It's easy to use and it will give you these optimized methods, basically, to have a optimal placement of windows to measure everything as good as it can be measured. So to sum it up a bit, we decided here to use an EvoSep system with a WISP before degradient, 15 centimeter ion optics columns, coupled it to a timsTOF ultra mass spectrometer, and for DIA analysis used optimized DIA analysis schemes designed with PyDI8. If we go now into the data analysis part, really, we're doing, of course, a DIA analysis, so we need to get a spectral library to analyze those samples. We're not the only lab that thought about doing DIA on immunopeptidomics. Of course, there are other publications like this top one, which goes into generating libraries for pooled libraries, and then later on analyze DIA samples from single sample that was part of the pool. Or also another publication really nice about benchmarking the bioinformatics pipelines for DIA analysis, which included de novo sequencing as an option to add more peptides to the library. But what both of these have in common is that they basically use measured libraries, so it's a really time extensive and sample extensive process. And as I said in the beginning, it's hard to get those samples. So what we thought about was using predicted spectral libraries. And what come into place here was the AlphaPepDeep package. It's a package spearheaded by a colleague of mine, Feng, who basically wrote the whole tool. And AlphaPepDeep at its core is a deep learning framework for spectral library prediction. So it was trained on a lot of peptide sequences and those properties, so like how they behave in a mass spectrometer, if you want to phrase it like that. And the deep learning framework was trained on it, and now it can predict peptide. If you provided a peptide sequence, it can predict the features, how it's gonna fly, how it's gonna fragment. So basically a spectral library prediction tool, which is also available on GitHub. You can just use it, download it, however you want to use it. You can test it out. It's nice because it's also adjustable to your mass spectrometry setup. It's not necessary to have a specific mass spectrometer to use this, but you can choose from a huge list of possible mass spectrometers and just predict your spectral libraries. Next to the spectral library prediction part, it also contains the PepDeep HLA package. And this really was key to do this library prediction for HLA peptides, because the PepDeep HLA package is basically a sequence prediction tool. So you take a large scale of HLA, public available HLA peptides, a large amount, and he trained a model which can distinguish HLA peptides from non-HLA peptides. So this model basically decides, okay, this peptide, from what I saw earlier with these millions of HLA peptides, looks similar to an HLA peptide, so it has a high chance to be in your sample in the end. You can tune this model by performing transfer learning with peptides that you suspect to be in your sample. This can be from prior measurements. This can be also from a direct DIA analysis. I'll show you in a slide how it looks like here. And this will basically tailor your model to your sample. This model then takes a FASTA file, whatever you give it. You can also give it a file that contains, I don't know, mutations or microbes or whatever. And it will basically select all the possible peptide sequences that the model decides have a high likelihood to be an HLA peptide. And then you end up with a sequence list, which you can then feed to AlphaPepDeep, which will then predict a spectral library for you, and you can use it for your DIA analysis. So in a bit more detail, we performed all the library-based searches with Diane, and for direct DIA searches, we use Spectronaut 16. So on the right-hand side, you see basically, again, a 200 microliter plasma sample measured on a 31-minute gradient with DDA. And if we now go on a scale from more experimental libraries to more computational libraries, a DDA-based library results already in an increase in peptide identifications, kind of as expected, also shown in the other publications beforehand. If we perform a direct DIA search on a DIA file, we end up with less peptides identified. This is usually because the search space handling for direct DIA searches is not tailored for open searches. So this results in a huge amount of possible peptides, which search engines usually have an issue with in terms of FDR control and really identifying peptides here. And if we now go to the non-tailored model, so basically a model which predicts all possible HLA peptides, so to say, from your FASTA sequence, we gain already identifications over the experimental library. And if we really now combine the model with our prior knowledge, so either from DDA-based search or from a DIA search, we really see a huge increase in identifications than what we can get in a single DDA analysis or even with an experimental library. As you can see here, the PAN library itself is already quite a good model, but you can really improve it with sample-specific knowledge here. The DDA-tuned library still outperforms the direct DDA-tuned library a bit, although I have to say the DDA-tuned library is trained on a single DDA measurement. So in my opinion, if you have a large cohort of samples or a large group of samples, the direct DDA-tuned library gives you already huge boost in terms of identifications, and this is really just a single measurement. So you can measure your whole sample, do a direct DIA search on it, tune your peptide HLA model with it, predict the spectral library, and then analyze the sample with it. And this really saves a lot of measurement time and also sample and cost here. So if we look now into the peptides that we identify, we can identify pretty many, I would say. So from just 200 microliters of sample, we can already get what previous publications aimed for with 5 to 10 milliliters of sample, which is basically the red line here. And we get a lot of peptides. I think it's around 30 to 40,000 peptides in this picture here from just measuring basically a day and a half of samples from just 6 different people. So it's really a huge benefit if you want to analyze a lot of samples, but also if you want to gain information about HLA epitopes, which are not really described in the common databases like the IEDB database, just because they weren't analyzed so far and the sample availability was limited. I also show here in light green 5 milliliters of plasma. These were fractionated samples, not injected in one. And these allow us really to go from 5 milliliters towards more than 10,000 immunopeptides for a single person. These are healthy donors and not cancerous donors, which gives you even, for cancer, usually have even more peptides. If you go to the properties, I said in the beginning, these peptides are usually all around 9 amino acids long, and we keep that feature. And also, if we compare soluble HLA peptides to their membrane-bound equivalents, we see that for the vast majority of HLA types, if we compare it to a membrane-bound HLA reference data set, we see a similar fraction of strong binding peptides. So doing this DIA-based spectral library, predicted spectral library-based analysis, but also analyzing soluble HLA peptides really gives us a good approximation of membrane-bound HLA peptides here. And if we now come back to the human where we had this sample from, it's really interesting to look into how do these peptides behave over time, but also what can we actually start comparing between people here. So if we look into a single person over the course of a year, we see a very strong correlation in terms of quantitative stability. So this is like one donor starting of the year, end of the year, and quantitatively very reproducible what we see in terms of the presented immunopeptides. If we now go towards a group of people, the picture is a bit different, but this is expected, right? Because at the beginning, I said the HLA type kind of guides what you present. And we have so many different HLA types within the population that two people having the same type or presenting the same thing is very unlikely. So also the quantitative correlation between peptides we find is supposedly very, very low. But if we look now into the qualitative similarity, we can already see that there are some people which share around 30, 40, 50% of the peptides. These are likely people that have a shared HLA type. And if we take this now to filter the previous picture, and look into the quantitative correlation of shared peptides between those people, we can see that we suddenly have a very high correlation between people, which is very interesting to look at, because this kind of tells, okay, there is kind of a mechanism of how these sHLA peptides are generated, and there could be kind of a ground truth between people, between healthy people and their sHLA peptidome. If we take those sHLA peptides now and we map them on top of a proteome data set, so we see another very interesting picture here. So this graph is a bit of a handful. I will take a minute to actually explain it in a bit more detail. What we did here was we basically mapped the peptides to the corresponding genes they belong to. And then we took a reference proteome data set, the genes from this proteome data set, and looked into, is this gene supposedly enriched for this organ? So what we assume here is that independent of how specific a gene or a protein is for a specific organ, as long as it is there, it has a similar likelihood to be presented by an HLA protein as all its counterparts. So this is basically an assumption, what we then transferred onto the immunopeptidome here. And what we basically see is that the frequency of those terms correlate really well between the soluble immunopeptidome and a proteome, so the organ proteome. So to put a picture to it, what we see here is that basically the duodenum has around 7.5% of genes which are assigned to be group enriched for the duodenum. And at the same time, we have a similar amount of peptides which are group enriched for the duodenum, which we find in plasma. So basically what this kind of looks like is that there's not a organ which produces all the immunopeptides in plasma, but more or less every organ that we had in this reference data set contributes to what we find in the soluble immunopeptidome in plasma. And this lets us really dig into organ-specific biology or organ-specific features by looking into the soluble immunopeptidome here. Because if we now go ahead and combine this with the previous analysis of shared peptides between individuals and correlating peptides between individuals, and we combine it with this assignment strategy here, what we can see is, for example, for, in this case, 3 people, comparing the intensity traces for immunopeptides. So each dot here is a immunopeptide assigned to a specific gene between those different people for, here in this case, the liver proteins, the selected amount of liver proteins that were sharely presented between these 3 peoples. And what is very interesting, that a lot of these genes are, the intensity traces are very similar, but for example, the cytochrome P oxygenases, CYP2A6 and CYP3A4, the intensities are very different between those 3 people. And this could likely be, for example, to lifestyle choices or diet choices. And this is very interesting that we kind of have a reflection in the soluble immunopeptidome towards what could maybe take place in the liver of that person at that time. And all of this is, of course, based on healthy donors, and it's a narrow amount of healthy donors, but it makes me really excited about applying this methodology to a bigger group of people and see if we see more of those effects that we can compare between people. And yeah. This already brings me a bit to the end of my presentation of this workflow and the strength of this workflow that I see. So what I really like about IMBAS is that it's high modularity. As I showed you in the beginning, you can do it automated, in this case with the Agilent Bravo system, but you can also do it on a lab bench, basically with just a magnet and your sample. You can just go ahead and dump the antibody in, put the beads in, and then just basically do a simple IP experiment. I showed you the results on an timsTOF ultra mass spectrometer, but you can also, of course, just exchange the mass spectrometer. Again, there you need to be really careful to adjust your method setup to immunopeptides and not just a standard triptych method here, because they're just really behaving differently. So other vendors are an option, other mass spectrometers are an option. What is also nice is that you can also do it with lysates or with tissues. You just have to add the lysis upfront. But you have to do that no matter what kind of immunopeptidomics enrichment workflow you're using. It's just exchanging really the antibody and beads here. And going towards a larger cohort of people, of course, is what I find very, very interesting. And I hope to see this also adapted and used much more to analyze really large groups of people, and really gives us a lot of more information about what is actually out there in the soluble immunopeptidome. On the bottom left, you have a QR code to the manuscript, basically. If you want to read more in detail about what I actually showed you, you can read it up all there, or you can just write me. And with this, I actually want to thank the group and Matthias Mann, who gives me the opportunity to do my PhD in this group. And a lot of my colleagues, most of them you see on this picture, not all of them, but I really want to thank all of them for their support and their help in doing this work here. And yeah, thank you for your attention, and I'm happy to answer questions.
Thank you so much, Maria. That was really a great talk. I see we have quite a few questions coming through in the Q&A. I think we can just run through them quickly. So I think you may have answered some of it, but we can just go through them anyway. Someone asked about the timsTOF platform and analyzing singly charged peptides. Do you want to comment on that?
Yeah, sure. So I kind of skipped the slide a bit too far. So there is a way to analyze the singly charged precursors on the timsTOF platform. If you do a DDA analysis, you usually apply a so-called threshold, like a filter, onto which ions are actually able to be selected for fragmentation and MS2 analysis. And if you adjust this filter to include also singly charged precursors, you can analyze singly charged precursors. Technically, you could leave away this filter, but this will also introduce all possible singly charged ions into your signal, which can end up really messy. So adjusting this filtering step is really what gives you the opportunity to also analyze singly charged precursors. There is another publication in MCP by Steve Carr's group that basically compared different DDA analysis, DDA method designs for the timsTOF and singly charged precursors. You can look it up there. It's a nice summary of different options and how to design those methods. And in DIA, of course, as I showed you, you can just adjust the method to include singly charged precursors.
Okay, and I guess leading on from your DDA explanation, now someone asked which method is better between the two, DDA or direct DIA? Or library-based DIA for that matter.
So, I mean, you saw the numbers for DDA
Yeah.
and direct DIA. If I just have the option to do direct DIA, I would do DDA, honestly, because you get more information here. I don't know if the if companies like Biochemistry, like Spectronaut, if they're gonna be an option for direct DIA for immunopeptidomics there. But you hardly just can do direct DIA, right? So if you can do a DIA analysis and you can do spectral library predictions for it, as I just showed, I would do the DIA analysis if your goal is to really have the highest depth of immunopeptides analyzed here. It's a bit of a it's an ongoing discussion, I would say, on what is really better here. What is important is to think about what do I actually want to do with those peptides? How can I validate those peptides? Because if you the spectral validation from DDA is still going to be cleaner than a DIA result. But if you say, okay, I can invest this and do the validation for peptides I identified by DIA, then I would go for a DIA analysis. But really the tools have to be in place to do that. If you can choose between DDA and direct DIA, I would still go towards DDA here. But if you can do what I just presented, then I think a DIA analysis can give you a lot of information from your sample here.
Okay. Somebody else says, hi, really amazing talk. Is there any intention to investigate the profiles of these neoantigens in different cancer types? It would be really nice to try and correlate this with clinical response and potentially move any leads into constructs for CAR-Ts.
I think this is more a question towards the community. I think there are intentions to actually do that. I mean, there are many more groups that work on immunopeptidomics than what I myself can do, and I think this is really the goal, what we can do with those peptides. And there, it also is really important to validate the peptides thoroughly before you go into a patient setup. I think this is actually done, or is actually what people do at the moment, which is what I'm very excited about to read in the future.
Okay. I see there's a whole bunch of questions. I hope we can get through them all, but if we don't, then we'll definitely try and answer them individually at a later stage. But let me try and summarize some of them into this similar theme. So someone says, the choice of the EvoSep was interesting. Did you try using any other LC systems? Were you concerned about losing peptides by going for the shorter gradients?
So the EvoSep system, I chose it because of the C18 chips, basically, because I didn't go into much detail here, but this allows the online cleanup, and having less steps really reduces the sample loss. I started those experiments on an EasyLC, basically, with dried down peptides, but in my hands, drying down really reduced, and doing the stage tipping up front and drying down really reduced a lot what I could measure from a single sample. So the EvoSep system was really the choice, because we have the C18 tips, we have this very high throughput that it enables, and also this really constantly running system that needs little maintenance. I can just put it in front of a mass spectrometer and start to run.
Okay. Another person says, great talk. And I think this is a really important question coming up now. Could you please elaborate about the blood collection and storage conditions, as well as the timeline with regard to sample prep? Because this can definitely have quite significant impacts on.
Yes. So I would say, although I don't have a very thorough analysis on this, like some publications do compare it in terms of plasma proteomics, the immunopeptidomics, I have probably less strong effects. I wouldn't say that there are no effects from different preparations, but they might be less strong than for plasma proteomics. But what I did here, what I showed you here is really plasma withdrawn in EDTA tubes. I directly cooled them down, or they were directly cooled, and then centrifuged. And the plasma was taken and then froze them in liquid nitrogen. It also works with serum. I didn't show it here, but a fecal serum preparation gives you a soluble immunopeptidome as well. There are differences between the serum and the plasma, but it works equally. It works equally well. These are the two methods I tested. The actual plasma protocol and how it was prepared is detailed in the manuscript. So centrifugation speeds, also timelines. For the plasma, I really took care of having it cooled down the whole time, and then froze it in liquid nitrogen. And then for the prep, it's just thawing them and performing the workflow. I wouldn't leave it standing in room temperature for long times, just because these IP protocols usually are better at 4 degrees, but yes, it still works if you leave it at room temperature, like the fecal prep basically shows, which is done in room temperature and not at 4 degrees.
Okay. What is the minimum amount of protein required for the enrichment of HLA peptides? Is 50 micrograms too less?
No, so if you go low enough with the sample input, you can use less than 50 micrograms. So for everything under 100 microliters of plasma, I basically use 1 microgram of antibody. And until After 200 microliters of plasma, I use 10 micrograms of antibody. It depends also, of course, on the purity of your antibody and what clone you're using. So I use the W632 antibody, which is like a commercially available and very widespread used antibody for HLA class 1 enrichment. And yeah, I mean, if you go to very high amounts, then you would need more antibody. But for a single sample, it's more than enough.
Okay, thank you. Another question, did you compare the method presented here with the cross-linked antibody? And if you did, how much better is the biotinylated method performing?
So I did perform some cross-linking experiments in the beginning, but what made me hesitant on continuing with those is really the potential errors while doing the cross-linking. In my opinion, if you have a perfect cross-linking experiment where you have not distorted or disoriented your antibody while cross-linking it, or over cross-linked it, or under cross-linked it, and you have the same antibody, just a different bead material, I don't expect a different result from a biotinylated antibody captured by the bead material. Here, really, for me, the ease of use is what made me choose the biotinylated antibody and the streptavidin beads. And I don't need experience with antibody cross-linking. I also don't need to to test my cross-links in the end, because I know my antibody is going to be intact. Yeah.
Okay. So we have two people who have asked which streptavidin beads exactly did you use, the normal, the Max, or MS?
I used the normal, if I remember it correctly. I think I also tested the Max. It worked as well. I didn't use the MS ones because I don't do a digest. And I mean, they are digestion protected. So I actually, yeah, I didn't test those because this was not really important for my protocol here.
I think there's a few more and more coming in.
No problem.
Are these soluble immunopeptides in the plasma, or are they coming from cells within the plasma?
So this is what I tried to show with the organ mapping here. There are a lot of very organ-specific genes that map to those immunopeptides and only map to those immunopeptides, or vice versa. These immunopeptides only map to those genes. And so the chance that they are from plasma cells is, I'd say very, very low. There could be potentially some that are also presented on cells that are in the blood, but I would say that are originating from tissues here.
Okay. Do you have any clinical metadata for the samples that you've analyzed, and did you correlate any of the findings?
So these were all healthy donors, so there's not really clinical metadata available for those.
Okay. And now the last two, if there's no other questions coming in. What did you use as the elution buffer? Was it 0.2% formic acid or 8 molar guanidinium hydrochloride?
When I set it up, I tested different elution buffers. Just GFA was one of them. These results are with glycine. So 200 millimolar glycine, pH 2. So it's a pretty standard elution buffer from adapted from other publications here.
Mm-hmm. And what was the ratio of the antibody to bead, or biotinylated antibody to bead?
So I, depending on the input amount, I use the amount of antibody, and especially for low amounts of volume, I really go to the lowest recommended amount of beads that to be used actually. So I think what is technically possible. If I go with 1 microgram of antibody, I can't really go lower in terms of the bead material usage here for what the beads actually are capable of. So I think it's like 10 microgram, if I remember it correctly. Again, it's written in the publication. I just do it day by day and I just pipette it and don't really think about those things anymore.
Okay, 2 more questions came in. Would you expect the biotinylated antibody to orient properly when coupled to the streptavidin beads, or is this antibody specifically engineered to be biotinylated at the FC region to ensure the right orientation? Okay.
It's the last one. Yeah, it's engineered to be only biotinylated at one specific position, because otherwise I would have the same problems as the cross-linking here.
Yeah. Someone says, great talk. Thank you for answering all the questions so far. How about single cell or DBP workflow for immunopeptidomics?
Yeah, I think that is a great challenge, and let's see if we can answer that question in the future.
So there's no more questions at the moment. Yeah, I think thank you again for this wonderful talk and for answering all of the questions so thoroughly. Oh, okay, someone just says thank you. Yeah, thank you, and thank you everyone for attending. Like I said, the recording will be made available in the next few days on the YouTube channel. If you want to rewatch it, if you want to go over the answers again, or if you want to share with your colleagues that couldn't attend. And thank you, Maria, again for this wonderful presentation. And thanks everyone for attending, and we'll see you again in the next one.
Thank you.
Yes.